Good afternoon, everyone. Welcome back to the second session of the fifth day of 1 week international webinar on the theme of Advances of Science in Education, jointly organized by Research and Consultancy Development Cell, Government Champhai College, and Mizoram Science, Technology and Innovation Council, MISTIC. Once again, I welcome you all. A good time to have in this session. And let me remind, if you have any questions or queries to the speakers, you can drop your question in the comment section, which is below, so that our speaker will clarify after the presentation. In this session, again, we have an eminent speaker, that is Dr. Laremruata Haunar. Before I give time to him, let me highlight his profile. Dr. Laremruata Haunar is an assistant professor at Government Champhai College in the Department of Zoology. He had done his Master of Science in Delhi University and PhD Life Sciences from School of Life Sciences, Jawaharlal Nehru University, Delhi. His research interest is epigenetic changes that influence the specific expression of certain genes from yeast to humans and the mechanism of environment influence on our genome, leading to the causation of genetic diseases. Dr. Laremruata Haunar had completed DST SERB Young Scientist Project from 2016 to 2019 and State Biotech Hub Mini Project from 2014 to 2017. He had 3 international research publications and 1 national publication as well. And today his title of presentation is on Advances in Epigenetics from Yeas to Mammals. Now may I welcome Dr. Laremruata Haunar to have his presentation.
Okay, thank you, Rebecca. Okay, from here, I'll start my talk. I'll share my screen first. It has been disabled, Maroda Sama Road. Can I share my screen?
Yes, you could share. You can go ahead.
Okay. Okay, thank you, Ms. Rebecca, for the kind introduction. So good afternoon, everyone. Today, the topic of my presentation will be advances in epigenetics from yeast to mammals. So epigenetics is a new field of science in biology, and it's not that new in history. It was started around 1940s, but it gained momentum during the 1990s. Then after that, the research on epigenetics has been blooming. So most of the work these days will be on epigenetics, especially in the field of molecular biology. So the talk will be on epigenetics, and I have divided my talk into 2 sections. The first section will be about yeast, the research conducted in yeast, and the next part will be on mammals, especially concerning the disease cancer. So what is epigenetics? So you need to know epigenetics. First, you need to know the structure of the chromosome. So this one is the chromosome, and chromosome is made up of chromatin fiber, and these chromatin fibers consist of proteins as well as DNA. So in the chromosome doesn't only consist of DNA, it also contains proteins, which plays an important role in the activation of the expression of the genes which are found in the DNA. And the units of chromatin fibers are called nucleosomes, and these are the nucleosomes. This one is one nucleosome, and this one is the next nucleosome. So nucleosome consists of histone proteins. 4 different types of histone proteins: H2A, H2B, H3, and H4. So these are the histone proteins, and you could see that there's DNA which is situated outside of the histone proteins. So because of this interaction between the histone proteins as well as the DNA, it causes a compaction of the chromatin fibers, leading to the formation of this compact chromosome. So epigenetics concerns with the changes which are found in the histone proteins as well as the DNA, and those specific changes causes a change in the expression of genes depending on the cell type, and those specific changes of expression could be inherited from the parent to the daughter cells. So that is the study of epigenetics. But one thing you should always remember is that epigenetics is very different from genetics, because genetics concern with the changes which are found in the DNA itself, but in epigenetics, most of the changes are found in the histone proteins, or some of them which modifies the DNA, but it doesn't change the sequence of the DNA. So in genetics, study of genetics, the DNA sequence are being changed or mutated, but in epigenetics it is not. So that is so epigenetics is like on top of genetics. So that is epigenetics. There are three very important modifications, or three types of modifications which are found in epigenetics. The first one is the histone modifications. As I have said before, this one is the nucleosome, which consists of the histone proteins as well as the DNA. So this is the nucleosome, and you could see that in these histone proteins, you could see different types of proteins, H through A, H through B, H3, and H4. And these are the N-terminal tails of the histone proteins. So you have a protein which is globular in nature or circular in shape, but there is the N-terminal tail of the protein protrudes outwards from the core histone, or the circular region of the histone proteins. So these are the N-terminal tails, and histone modifications occurs on the tails of these histone proteins. So these modifications are the these modifications are the ones that causes changes in the compaction of the chromatin fibers. Some of the changes which occurs on the N-terminal tails are the methylation, acetylation, and ubiquitination. So these histone terminal tails, they consist of amino acids, and those amino acids could be modified using methylation, acetylation, phosphorylation, or ubiquitination. And then the second one is DNA methylation, where methyl group is attached to the cytosine residues of the DNA. So this change or this modification does not causes a change in the sequence of the DNA, but it merely attaches methyl group onto the cytosine residue only. And this addition of methyl group is very important as it controls gene expression. And the third one is the chromatin remodeling. In chromatin remodeling, the chromatin remodelers use ATP so that these histone proteins is removed from the DNA, so that you have a naked DNA, and that naked DNA will be available for expression of genes. So these epigenetic modifications, the three of them, are involved in the expression or overexpression or underexpression or repression of the genes. So why does all these epigenetifications occur? And they occur because of the environment. Sometimes you used to take drugs, medicines, and sometimes you used to eat food also, depending on the food, as well as the lifestyle that we have, the amount of sleep that we have, or the amount of exercise that we used to take. Those are the environmental changes that causes epigenetic modifications in our body that leads to the changes in gene expression, so that the phenotype, whether we are tall or small or big or fat, like that. So those things are being controlled by these epigenetic modifications. So genetics also controls the phenotypes, but even epigenetics, which is caused by the environment or influenced by the environment, can causes phenotypic change in our body. So some of the characters, important characters, could also be changed using our environment. And epigenetics is very important because it acts as an on or off switch, on or off switch in our genome, because we have around 20,000 genes in our body or in our genome, and all those genes are not expressed at one time. Okay, so they are not expressed at one time, but only around 10 to 15% are being expressed in a certain cell or in a specific cell, and that occurs because of these epigenetic modifications, because epigenetic modifications act as an on or off switch. So in a certain cells, like in neurons, some proteins, the uh, mm, the neuronal proteins are being expressed because of these epigenetic modifications. But in the blood cells or in the skin cells, the neuronal proteins are not being expressed, okay, because of these epigenetic modifications. So epigenetics is very important in the study of molecular biology. First, we'll talk about the role of histone acetylation in yeast. As I said before, histone acetylation is a part of the histone modifications, which we had talked about in the slides, a few slides back. And particularly, we had done our experiment in Candida albicans, which is a species of yeast. And this Candida albicans is a dimorphic fungus which causes infections in our body, and those infections could be life-threatening if the patient is immunodeficient, like in AIDS or cancer patients who had undergone chemotherapy, or those people who had undergone organ transplantation. They used to have a weak immune system. So if they have those weak immune system, and if there is Candida infection, it could lead to the death of the patient. So that is how vital it is to study Candida albicans, and so that new drugs could be used for their treatment. And these Candida albicans could exist in three forms. The first one is the budding form or the yeast form, which is circular in shape. And the next one is the pseudo-hyphal form. This one is a little bit elongated. And then you have the hyphal form. And this hyphal form is the elongated form of the yeast form, where you could see that there is an elongated structure. You could see the elongated structure of the Candida albicans. And this hyphal form is very important for the infections to occur in our body, because this hyphal form could penetrate our tissues, or it could penetrate into the blood, so that infections could occur. Okay, so this the ability to change its form from the yeast form to the hyphal form is very important. And so we did our studies and look at and check how histone acetylation could affect the formation of this hyphal form. So first we need to talk about the histone acetylation in Candida albicans. And histone acetylation, it simply is an attachment of acetyl group into the histone proteins. As I have said before, the histone or the core histone proteins have N-terminal tails, which consists of amino acids. And those amino acids, most of them are lysine residues. And in those lysine residues, this acetyl group is attached to the lysine residues. So that is called histone acetylation. And this histone acetylation is very important because it results in the gene activation of a particular gene. So it is one of the most important transcription factors that we have found. And this reaction is catalyzed by an enzyme called histone acetyltransferases. So histone acetyltransferases are the enzyme which transfers the acetyl group from acetyl CoA onto the histone proteins. And so the reason why it activates gene gene expression is because this acetyl group has negative charge and a negative charge. And we know that the phosphate backbone of the DNA is also negatively charged. So if two negative charges comes together, they used to repel each other. So with the histone acetylation, you could see that there is a stranded portion of DNA or naked portion of DNA which could be used for histone acetylation. So that is how uh, histone acetylation affects transcription or expression of a certain gene. In Candida albicans, the main histone acetylating enzyme is called GCN5, and this GCN5 uh, results in if it acetylates the genome or the promoter of a gene, it results in an open formation of open chromatin structure. So the chromatin structure will be opened up, resulting in the expression of genes because transcription factors could bind to the open chromatin structure. So this Candida Candida albicans it is diploid in nature, meaning it has two copies of a gene. In our studies, what we had done is we knock out the true copies of GCN5, the one which is the histone acetylating enzyme, two copies of GCN5 from the genome of these Candida albicans, and we found that there is no expression of GCN5, so resulting in lower global histone acetylation. And then after that, we check whether the is2 hyphal formation is affected or not. Okay, so we knock out the gene using homologous recombination technique. It's an easy technique. And very efficient also. So with the deletion of GCN5, we look at how the is2 hyphal formation has been affected. And we came to find out that when you knock out or delete the GCN5 in Candida albicans, it leads to reduced hyphal formation. This one is the wild type, BWP17 is the wild type, and this one is the null mutant, where true copies of the GCN5 are being deleted or knocked out. As you can see in the wild type, with the hyphal induction, you could see the formation of hyphal form. You could see all these elongated form of the Candida albicans. This one is the hyphal formation, which is found in the wild type. But when you knock out GCN5 in the Candida albicans or in the wild type, you could see that you could see all the circular structure or the yeast form, and you could barely see some hyphal form or pseudo hyphal form found in the null mutant. This shows that GCN5 is important in hyphal formation. And we count the number of cells. We count at least 1,000 cells and found that the percentage of cells which shows hyphal form in the wild type is around 75%, which is around 75% and very high. Uh, and we also found that the yeast form or the circular form, the non-pathogenic form, is very less in the wild type, which is around or which is lower than 20%. And if you look at the null mutant, you could see that the hyphal form, the percentage of cells forming hyphae is very less, which is still lower than 20%. But the amount of cells which show the circular form or the yeast form, the non-pathogenic form, is very high, which is around 80, almost 80%. So we can conclude, or we can just assume that deletion of GCN5 is very critical in the hyphal formation of Candida albicans. To form hyphal, to change its form into the hyphal form, there are two very important transcription factors. And those two transcription factors are EFG1, this one, EFG1, and CPH1. These two transcription factors are very important for the formation of hyphae because they are the ones that that causes the expression of hyphal proteins. So without hyphal proteins, you could not have the hyphal form. And for the formation of hyphal proteins, you need EFG1 you need EFG1 and CPH1. So these two transcription factors are very important. And this EFG1 is part of the, it acts downstream of the cyclic AMP PKA pathway, and CPH1 also acts downstream of the MEP kinase pathway. And these two pathway, the cyclic AMP PKA pathway and the MEP kinase pathway, acts downstream of another regulator called RAS1. And this RAS1 is the master regulator of transcription in Candida albicans. It is very important because it controls most of the transcription factors in Candida albicans. EFG1 and CPH1 also acts downstream of the they also act downstream of the RAS1. So we in our next studies, we know that the hyphal formation has been compromised in our mutant, but we don't know the mechanism. So to look at the mechanism, how this occurs, we check the expression level of these two transcription factors, EFG1 and CPH1. We came to find out that the transcription level or the expression level of the two transcription factor 1 and CPH1 is very low, which is very, there's a significant reduction in their expression. So that probably is the reason why hyphal formation is affected. And as I have said before, this EFG1 and CPH1 acts downstream of RAS1. So we look at the RAS1 expression also using real-time PCR, and we came to find out that the mRNA level or the expression level of RAS1 is very less, which is around 50% down. So there's a significant reduction in the RAS1 transcription level. So with this reduction in RAS1, the RAS1 protein, so there is very less RAS1 protein in the null mutant. So those significant reduction in the RAS1 protein probably causes the significant reduction in the expression level of EFG1 and CPH1, ultimately leading to the reduced expression of hyphal proteins. So the hyphal formation in our null mutant is affected because of the RAS1 expression, which is being controlled by our gene GCN5. So GCN5 controls the expression level or the expression of the RAS1 protein, ultimately leading to the control of the hyphal proteins. So indirectly, GCN5 controls the expression of the hyphal proteins, which are necessary for hyphal formation. So how does this histone acetylation affect the expression or synthesis of transcription factor RAS1? Now we know that GCN5 affect the RAS1 expression, but we don't know how it affects or how this GCN5 controls RAS1 expression. So what we had done is that we look at the binding of RNA polymerase II. RNA polymerase II is very important because it is the actual enzyme that produces mRNA from DNA. So we know that for an expression of a gene, the DNA has to be converted into RNA and then into protein. So first RNA has to be made. And RNA polymerase II is the enzyme that produces the mRNA from DNA so that protein synthesis could occur. Okay, so and for this RNA polymerase to do its function, it has to bind to the promoter of a certain gene. Like in the case of RAS1, it has to bind to the promoter. It has to bind to the promoter of the RAS1 gene so that it could induce the expression of the RAS1 gene. So we did chromatin immunoprecipitation experiment, and we found out that the binding capacity or the amount of RNA polymerase II which binds to the RAS1 promoter is very less, and it is down by around 60%. So around 60 to 70%. So if there is less RNA polymerase II binding onto the binding onto the promoter of the RAS1 that will ultimately lead to reduced expression of RAS1. So that is how GCN5 works. GCN5 with the inarnal mutant, with the deletion of GCN5 inarnal mutant, that leads to reduction in global acetylation of the genome. It affects the binding of RNA polymerase 2, especially in the RAS1 promoter. So since there is no RNA polymerase 2 to cause the expression of RAS1 protein, leading to reduced hyphal formation in our null mutant. So to summarize the first part of our work, the knockout or deletion of GCN5 leads to reduced histone acetylation, including the RAS1 promoter, resulting in close chromatin complex on RAS1 promoter. So with the formation of close chromatin complex on RAS1 promoter, it leads to reduced RNA polymerase 2 binding, resulting in lower expression of RAS1 protein. So ultimately, it leads to reduced hyphal formation in the null mutants of GCN5. So what our experiment or what our work has shown is that histone acetylation is important for the induction of hyphal formation in Candida albicans, and GCN5 seems to be an important regulator for the switch between hyphae and Yeas form. So as I said before, the switch between hyphae and the Yeas form is very important for the invasion or the for invasion into the tissues or the human, as well as for its virulence. So GCN5 seems to be the switch that could switch that could decide between the hyphal form and the Yeas form. And this GCN5 could be a drug target in fighting against the candidiasis or candida infection, especially immunodeficient patients like AIDS and cancer patients. So this is the first part of my presentation where I had talked about histone acetylation. Next, I'll talk about the role of DNA methylation in gastric cancers. Gastric cancer, it is the second most common cancer in the world, and it is the fourth. I think it is the fourth in number when it comes to cancer-related death. So it is very important to study gastric cancer, and DNA methylation has played an important role in the development of gastric cancer. So as I said before, in the first few slides, DNA methylation is a part of the epigenetic modifications, where you could see that this one is the cytosine, and in this cytosine, a methyl group is attached to the cytosine, causing changes and resulting in a formation of 5-methyl cytosine. So this one is called DNA methylation, and it is catalyzed by DNA methyltransferase. And there are 3 enzymes: DNMT, 3A, 3B, as well as DNMT1. So these are the enzymes which causes the DNA methylation. And this DNA methylation is very important in the expression of genes. As I said before in the last slide, we have a promoter where it is the site where the RNA polymerase binds, and other transcription factors also bind to cause expression of a certain gene or a specific gene. So you have this one is the RNA polymerase, and you have all the other transcription factors which are necessary for the expression of a gene. And this RNA polymerase cannot function without the other transcription factors also. So they all bind to the promoter with the RNA polymerase 2 and causes transcription of a specific gene. In our genome, most of the promoters, around 60% of the promoters, are tatalase promoter, and this tatalase promoter contains are rich in CpG dinucleotides. So most of the promoters contains CpG dinucleotides, and they are called as CpG islands. And these CpG dinucleotides are the targets for this DNA methylation, because DNA methylation to the cytosine residue occurs only when the cytosine residues is next to guanine residues. So this CpG dinucleotide is very important, and it is most of the time it is found in the CpG island or in the catalase promoters of a gene. And so if this methylation occurs through the promoter, then it could lead to reduction in gene expression, because if methylation occurs to the promoter, then it recruits proteins other than these transcription factors. So with the recruitment of other proteins, that leads to the that blocks the binding of the RNA polymerase II as well as the transcription factors. So that is the importance of DNA methylation in our body. So if there is any DNA methylation in the promoter, it will lead to the repression of a certain genes. And there has been so many works going on in the role of DNA methylation in cancer, especially in the stomach or gastric cancer, where they had found that there are two methylation events which occur during gastric cancer. The first one is the global methylation. In this global hypomethylation, the methylation status of most of the promoters are being released. In a sense, the methylation which occurs on the catalase promoters are being relieved or removed, such that it results in the activation of proto-oncogene and genomic instability. Oncogenes or proto-oncogenes are the ones which, when mutated or upregulated, leads to the formation of cancer, because they are the ones that causes or that induces cell growth and cell proliferation. And so with the global hypomethylation, it leads to activation of the proto-oncogenes. And so if the proto-oncogenes are being upregulated, then they will cause uncontrolled cell proliferation, leading to cancer. And the global hypomethylation also causes genomic instability, because we have lots of long non-coding RNAs, long non-coding RNA like line 1 and ALU sequences in our genome, and those sequences are being repressed using methylation. But if this global hypomethylation occurs, then those long non-coding RNAs will be activated, leading to addition or deletion of certain chromosomes, resulting in genomic instability, which is found in cancers. And the other methylation event, which is found in gastric cancer, is focal hypermethylation. So, well, the methylation status of the cell may be down, but there is specific hypermethylation which occurs in gastric cancers, which turns off turning off the tumor suppressor genes. There are certain genes in our body, tumor suppressor genes, that repress the formation of cancer. So these are called tumor suppressor genes, and their products are very important because they're the ones that tries to repair the DNA or tries to inhibit the oncogenes, these proto-oncogenes, so that cancer formation does not occur. But because of focal hypermethylation, they are being turned off such that it leads to the formation of cancer. So this is how DNA methylation works in cancer. As I've said before, we have two very important different types of genes. First one is the proto-oncogene, and the second one is the tumor suppressor genes. We'll look at 3 tumor suppressor genes and 1 proto-oncogenes in the gastric cancer. And the first one is the CDH1 tumor suppressor gene. This CDH1 tumor suppressor gene is found in all the cells, and it encodes a protein called E-cadherin. And this E-cadherin is the protein which is involved in cell-to-cell adhesion. It is involved in cell-to-cell adhesion, and this E-cadherin, the product of CDH1 gene, is activates Wnt and not signaling pathway, which are involved in cell proliferation, and it also controls the mTOR signaling pathway, which can lead to cell growth as well as cell division in our body. So those pathways are being controlled by CDH1, and what they had found is that people have done research and they have checked the methylation status of CDH1 in gastric cancer patients, and they had found that in around 50% of the gastric cancer patients, they have found that the CDH1 promoter had undergone methylation. So there is hypermethylation in the CDH1 promoter, leading to reduced expression of E-cadherin, and that ultimately leads to the formation of gastric cancer. So methylation is very important, especially in the case of tumor suppressor genes, which prevents us from developing cancer. And this CDH1 is very important because it is this gene which once it mutates, undergoes mutation or promoted methylation, then it leads to cancer. So this one is the first step, the most important step in controlling the formation of gastric cancer in our body. And the next one is the protein called amylase-1. This one is amylase-1, which is involved in DNA mismatch repair pathways. So we all know that during DNA replication, mismatch occurs sometimes. So if those mismatch mutation occurs, if they are not repaired, then it could lead to accumulation of DNA mutation such that it leads to mutation of a certain gene. And if that gene is if it is an oncogene or tumor suppressor gene, it could lead to the formation of cancers. So to prevent those formation of mismatch, this amylase-1 is very important because this amylase-1 identifies the mismatch during DNA replication, and with the identification of the mismatch, it leads to the repair of the mismatch. So it serves an important functions in our body, especially in our genome. So these people had checked the methylation status of promoter methylation status of the amylase-1, and they found out that in around 50% of the gastric cancer patients, there's a promoter methylation of amylase-1. So in this 50% of the gastric cancer patients, if amylase-1 expression does not occur, then that could lead to the accumulation of mutations or mismatch in our genome that ultimately leads to the mutation of a certain gene, some of the genes like the tumor suppressor gene or the oncogenes, leading to the formation of gastric cancers. So this is the one that prevents gastric cancers in our body. And if this one is repressed or underexpressed, that could lead to gastric cancer in the body. So another tumor suppressor gene that I would like to talk about is CDKN2A. So this one is a cell cycle regulator, and it is involved in cell cycle regulation because this gene CDKN2A encodes a protein called P16, and this P16 protein inhibits the activity of CDK4/6 and cyclin D. And this complex is very important because it drives the cell cycle or the cell proliferation. So if there is cell proliferation, uncontrolled cell proliferation, then it could lead to the formation of cancer. So to prevent that, we have CDKN2A, which encodes the protein P16. So what these people have done is that they check the promoter methylation of CDKN2A in Caucasian, Asian, and others. And what they have found is that they have around 3,067 samples. They take samples from 3,067 people. And what they have found is that when they have checked the methylation, promoter methylation of the patients, gastric cancer patients, they found that the promoter of CDK and 2A had undergone methylation. So which means that in around 50 to 60% of the gastric cancer patients, this CDK and 2A does not work. So if it doesn't work, then it leads to cancer. So that is how important methylation, epigenetic modification is in the formation of gastric cancer patients. And lastly, I just want to talk about the methylation status in the oncogene called CMYC, transcription factors, which is very important because it drives cell proliferation, and it is very active during during embryo formation as well as in the development of the fetus. But once it is activated during adulthood, then it could lead to the formation of cancer because it drives cell proliferation or cell division. So these people have checked the promoter methylation status of CMYC, and what they have found is that the promoter of the CMYC has been unmethylated in almost all of the patients. So they have taken gastric cancer patients, and you could see that U represents unmethylated status, and M for the methylated status. So they have what they have found out is that they have taken four samples from gastric cancer patients, and what they have found is that in all the four patients, the promoter of the CMYC had undergone unmethylation. So there is no methylation to repress the expression of CMYC. As I have said before, if this CMYC is overexpressed during adulthood, then it could lead to cancer. So what they have found is very important because in gastric cancer patients, CMYC has been overexpressed due to the hypomethylation of their promoter. So that probably leads to the formation of gastric cancer and its progression in the body. So this is how important the promoter methylation of tumor suppressor genes and oncogenes is in the development of gastric cancer. In the first few slides, I had talked about histone modifications. And in those histone modifications, there is acetylation also, there is methylation, and there are so many other important modifications also like that, chromatin remodelers. And those modifications are also found to be involved in gastric cancers. And so gastric cancer, as we can say, is that even though genetic mutation might happen, but even epigenetic modification or epigenetic mutations can also cause gastric cancer in our body. That's how important epigenetic modification is. Okay, and so let's look at the picture, the gastric cancer picture in Mizoram. And we all know that Mizoram is the cancer capital of India, and Mizoram has the highest number of cancer patients due to, according to the Economic Times in 2015, where Dr. Eric had talked about the number of cancer patients, and he had said that there's a very high incidence of stomach, esophagus, lung, and cervix cancer in Mizoram. And according to the population, Mizoram leads the world in these four types of cancer. So Mizoram has a very high incidence of cancer if you look at the total number of population or the number of people, and the average death toll due to cancer per year is 711 in Mizoram in 2019. So 711 people died because of cancer in Mizoram in 2019 alone, and there are currently 710 cases of stomach cancer, which is the most common in Mizoram, and followed by lung cancer at 688 cases. So Mizoram, even though it is a very small state, still leads the world when it comes to cancer. So this has led to uh, this has led us to fight against cancer, especially among the research scholars. And so Dr. Eric and others have done their work, and they have found that the peculiar food habits which is associated with the Mizo people seems to be the prevalent cause of stomach cancer in Mizoram. And they have also worked that extensive consumption of tobacco coupled with the consumption of smoked meat leads to high incidence of cancer. So most of their work had shown that the dietary habits that we had seems to be the main reason for the development of cancer in Mizoram. But the groundwork is there. These are all epidemiological studies, but there are no experimental studies or experimental research which had been done in Mizoram. So epigenetics could provide a very good way of studying how prone we are, or how easy for the Mizo people to get cancers, especially to get cancer or develop cancers, because as I've said before, Mizoram has the highest number of cancer patients in the world. So epigenetics could provide a new way to fight against cancer in Mizoram. As I have said before, people have shown that whatever we do, the things that we eat, or whether we used to take exercise, or the amount of sleep that we have, the lifestyle that we have, all have an impact in our epigenome. So the epigenetic modifications that we had talked about, the methylation, the histone acetylation, or any other modifications that we had talked about, are being caused by our environment. So Mizoram has a very different dietary habits with different lifestyle, and that lifestyle could change the epigenome in our body, leading to the development of or high incidence of cancer in our state. So epigenetics could provide a new way to find out how prone we are to cancer, as well as to fight against cancer. That would be a very good way to start our battle against cancer. So that will be for the future work. Okay, that will be it for my talk, and thank you. And this is the Oncology and Epigenetics Lab in Department of Biotechnology, MZU, and these people are the ones that help me in our work. Okay, thank you so much.
Thank you, sir, for your interesting and information presentation. Our patient has come and enlightening us with your knowledge. Also, I would like to thank all the participants for your patience, dedications, and sparing your time. I hope that the presentations would be fruitful to each one of us. With that, we end up our session. Thank you all.